Dear viewers, greetings. In this video, let's see about the human parainfluenza virus. Introduction. Human parainfluenza viruses are a group of viruses that cause different types of respiratory infection and are most common in children and babies. Most human parainfluenza viruses usually cause infection of the upper airway such as common cold, ear infection or sore throat. Other infections caused by human parainfluenza virus include infection of the lower respiratory tract such as croup, pneumonia or bronchitis. Structure of human parainfluenza virus. It belongs to the family of Paramyxoviridae. Parainfluenza virus resemble orthomyxoviruses in morphology but are larger and pleomorphic. The size ranges from 150 to 300 nanometer and the symmetry of parainfluenza virus is helical. It is an enveloped virus and the genome of parainfluenza virus is negative sense single-stranded RNA virus. Types of human parainfluenza virus. There are 4 types of human parainfluenza virus. Type 1 and 3 belong to the genus Respirovirus and type 2, 4A and 4B belongs to the genus Rubella virus. These 4 types of parainfluenza virus are most common associated with cold-like symptoms. Parainfluenza 1 and 2 are cold-like symptoms and parainfluenza virus 3 and 4 usually cause mild to severe illness. The 4 types of parainfluenza viruses are type 1 Sendai virus, type 2 acute laryngotracheobronchitis, type 3 respiratory infection in children and type 4 respiratory infection. The transmission of human parainfluenza virus is that it spreads from person to person contact through the respiratory droplets. The incubation period is 2 to 6 days. Replication. The replication strategy is that it belongs to the negative stranded RNA virus. These viruses replicate entirely within the cytoplasm. Viruses dock to the cell using glycoprotein or glycolipid cell receptor and its own GH or HN spike. The virion enter the cell by mediating the fusion of the lipid envelope, and this vital event of catalyzed fusion is mediated by F protein. The genome is released immediately into the cytoplasm. The genome is transcribed by viral RNA transcriptase to give subgenomic, generally monocystronic mRNA. Shortly afterwards, gene transcription switches into gene replication when full-length positive RNA strands are synthesized, which act as a template for RNA replication. The newly synthesized negative RNA strand interact with the N protein and the virion transcriptase, and these nucleocapsid structure and then combine with viral M protein. The viral spike already inserted into the plasma membrane to the cell interact with the M protein. The virus are released from the cell by budding. Pathogenesis of parainfluenza viruses. Parainfluenza viruses infect the epithelial cells of the upper respiratory tract. Then the virus replicate more rapidly than measles and mumps virus and can cause giant cell formation and cell lysis. Unlike measles and mumps, the parainfluenza viruses rarely cause viremia. The virus usually stay in the upper respiratory tract, causing only cold-like symptom. In approximately 25% of the cases, the virus spreads to lower respiratory tract, and in 2 to 3% of the disease may take the severe form of laryngotracheobronchitis. Clinical manifestation of human parainfluenza viruses. A parainfluenza virus in infant may be more severe than the infection in adults, causing bronchiolitis, pneumonia, and most notably croup, that is laryngotracheobronchitis. Group results in subglottal swelling that may close the airway. Parainfluenza viruses 1 and 2 and 3 may cause the respiratory tract syndrome ranging from mild cold-like upper respiratory tract infection, that is coryza, pharyngitis, mild bronchitis, wheezing and fever, to bronchiolitis and pneumonia. Older children and adults generally experience milder infection than those seen in young children, although pneumonia may occur in the elder persons. Most children recover within 48 hours. The symptoms of parainfluenza virus include common cold, nasal congestion, runny nose, sore throat, cough, inflammation of larynx and upper airway, fever, inspiratory stridor, intercostal retraction, diffuse inflammation with erythema and edema in the tracheal walls, coryza, pharyngitis and wheezing. Laboratory diagnosis of human parainfluenza virus. The specimens may be taken from buccal or oral swab, saliva and urine. Direct immunofluorescence test and heme adsorption inhibition test are used for the antigenic detection of the virus. Primary monkey kidney cell and shell viral technique are used for the isolation of this parainfluenza viruses. Cytopathic effect includes cell rounding and giant cell formation. ELISA neutralization test and heme agglutination inhibition tests are used for the serum antibody, and to detect the viral RNA, RT-PCR is most commonly and widely used method. Treatment of group consists of the administration of nebulized cold or hot steam and be carefully monitoring of the upper airway. On rare occasion, incubation may become necessary. No specific antiviral agents are available for the human parainfluenza viruses. Vaccination with killed vaccine is ineffective and possibly because they fail to induce the local secretory antibody and appropriate cellular immunity. No live attenuated vaccine is available. References are taken from Medical Microbiology by Murray 8th edition and Basic Virology by Edward K. Wagner, Martinez J. Thank you.